I'm Professor Steve Jackson at the Gurdon Institute, part of the University of Cambridge. My research studies how cells respond to DNA damage and what goes wrong in certain diseases, such as ataxia telangiectasia, which is underpinned by a defect in DNA repair. With previous funding, we've obtained cells from a very special AT patient who has a very mild progression of the disease. And what we want to study from the cells from that patient is to see whether or not there's something special about the background of that patient, the genetic background, which makes those cells and therefore the person resilient to the disease. So with previous funding, we've managed to obtain samples from that patient and derive cells called iPS cells. These are induced pluripotent stem cells. These are very special cells because we can grow them in the lab here and we can differentiate them into various cell types, including neurons. And that's important because one of the most debilitating aspects of AT is loss of neurons in the brain, which leads to the coordination difficulties of the disease. So what we're doing here together, my lab together with the lab of Dr. Rick Livesey here in the Gurdon Institute, is to grow those cells. We've now got them growing, and what we want to do in our future research is to differentiate those into neurons and therefore study the underlying mechanisms that we think may be of great importance for AT as a disease. With the funds kindly provided by Action for AT and the AT Children's Project, we now propose to build on our previous research to take this into exciting new dimensions. So first of all, we're taking the iPS cells that we've already established and we're going to differentiate those into neurons in cell culture and study those neurons either derived from normal individuals, patients with full-blown AT, that's the full disease, or cells derived from this patient with mild AT, to see if we can identify any differences between them which might explain how the disease arises and rises at different rates, if you like, in the individuals. Secondly, we're going to combine those studies with an additional dimension of work, which is based on some genetic screens that my lab has carried out. So in brief, what we've done recently is to take AT cells, that's cells lacking ATM, and we've carried out a so-called CRISPR-Cas9 genome-wide screen to look for other genes that, when knocked out, alleviate the cellular phenotypes of AT. That's been very successful. We've already identified certain genes that when they are knocked out, they alleviate the cellular DNA repair symptoms, pathologies if you like, of these AT cells. And what we want to now try and do is combine that study with the studies of patient-derived cells to see whether or not those same genes or other genes we might identify might alleviate the cellular phenotypes, the cellular properties of these AT cells. So work in my lab is fundamental cell and molecular biology. We're trying to understand the basic cellular mechanisms. But it's always been my belief, and I guess the belief of many others, that if you understand precise cellular mechanisms and can understand what cellular mechanisms go wrong in a disease, such as AT, that can provide insights into the disease. And from that understanding, we might be able to come up with new therapeutic opportunities. And in a more specific way, if we can identify genes which modify, which affect the disease severity at the cellular level, the hope will be that those same genes will affect severity at the level of the patients. And if that's the case, we can imagine an opportunity potentially to drug those targets and potentially develop drugs that might slow down, ameliorate the progression of AT. That's the long-term hope of this type of basic research. Well, I think there are a number of reasons why people should support charities such as Action for AT and AT Children's Project. First of all, these charities are established to try and ultimately improve the lives for young people who have this devastating disease. And it's my belief that out of basic research into understanding disease mechanisms will ultimately arise better treatments for these diseases. Secondly, this research has broader ramifications, because if you can understand the basis of a rare genetic disease such as AT, that can also provide you major insights into, for example, other genetic diseases which may share similar pathologies, and potentially can also provide insights that could be useful to understand neurodegenerative disease, for example, or cancer in the wider population. So I think rare disease charities and the research that they fund isn't just of relevance to those patients, it has much broader potential.